Welcome to NEET Biology MCQs educational channel. Are you a NEET aspirant? Get ready to challenge your brain with high-quality MCQs based on the NCERT book. Sharpen your concepts, enhance application skills, and master assertion reasoning and statement-based questions. This is your ultimate MCQ power zone for NEET success. To prepare a pollen smear from hibiscus, what should be the first step? A. Collect stigma. B. Crush the ovary. C. Open anthers and sprinkle on slide. D. Boil flower buds. Correct answer C. Open anthers and sprinkle on slide. Explanation. Mature anthers contain ready-to-release pollen. Gently opening and tapping them over a slide is the proper technique. The stigma, A, and ovary, B, contain female parts. Boiling buds, D, would destroy pollen structure. Which wall layer of the anther directly nourishes developing pollen grains? A. Epidermis. B. Endothecium. C. Middle layers. D. Tapetum. Correct answer is D. Tapetum. The correct answer is D. Tapetum. The tapetum is the innermost wall layer of the anther and plays a vital role in providing nutrients and enzymes necessary for the development of microspores, pollen grains. It is metabolically active and often has multinucleate cells. The epidermis, A, is the outermost protective layer and does not contribute to nourishment. The endothecium, B, helps in anther dehiscence by generating tension, but it doesn't nourish pollen. The middle layers, C, degenerate early and have minimal contribution to nutrition. Hence, only the tapetum serves the nutritive function for developing pollen grains. What makes the exine of pollen grains highly durable? A. Cellulose. B. Pectin. C. Sporopollenin. D. Lignin. Correct answer is C. Sporopollenin. Explanation. The correct answer is C. Sporopollenin. The exine layer of pollen grains is made up of sporopollenin, a complex polymer known for being chemically inert and highly resistant to physical and biological degradation. This makes the exine extremely durable, allowing pollen grains to withstand harsh environmental conditions and even persist in fossil form. Cellulose, A, and pectin, B, are found in general plant cell walls but are not responsible for this kind of durability. Lignin, D, adds rigidity to vascular tissues, not pollen grains. Thus, only sporopollenin is associated with the exceptional resilience of pollen exine. How many cells are present in mature pollen of most angiosperms? A. 1. B. 2. C. 3. D. 4. Correct answer is B. 2. Explanation. The correct answer is B. 2. In most angiosperms, pollen grains are released from the anther in a two-celled stage, consisting of one vegetative, tube, cell and one generative cell. The generative cell later divides to form two male gametes. Option A, 1, is incorrect because a single-cell stage occurs before maturation. Option C, 3, is incorrect in this context, though some species release pollen in the three-cell stage, vegetative cell plus two sperm cells, but it's not the majority. Option D, 4, refers to the tetrad stage during microspore formation, not mature pollen. Therefore, two cells are typical in mature pollen for most angiosperms. Where are microsporangia located in the anther? A. On the filament. B. At the tip of style. C. In the corners of each lobe. D. At the base of stigma. Correct answer is C, in the corners of each lobe. Explanation. The correct answer is C, in the corners of each lobe. Each anther lobe, there are typically 2, contains 2 microsporangia located at its corners, making a total of 4 per anther. These are the sites where pollen develops. Option A, filament, is incorrect because the filament is the stalk that supports the anther but does not contain microsporangia. Option B, tip of style, and D, base of stigma, are female floral structures, not related to microsporangia. Thus, only option C correctly identifies the location of microsporangia. What determines how long pollen grains remain viable? A. DNA structure. B. Temperature and humidity. C. Number of pores. D. Flower color. Correct answer is B. Temperature and humidity. Explanation. The correct answer is B. Temperature and humidity. The viability of pollen grains, how long they can function and fertilize an ovule, varies with environmental conditions. High humidity and moderate temperature generally enhance viability, while dry or extremely hot conditions reduce it. Option A, DNA structure, plays no role in short-term viability. Option C, number of pores, influences pollen tube emergence but not lifespan. Option D, flower color, is entirely unrelated to pollen viability. Therefore, external environmental factors are the primary determinants. Which among these families shows long pollen viability? A. Solanaceae. B. Poaceae. C. Liliaceae. D. Orchidaceae. Correct answer is A. Solanaceae. Explanation. The correct answer is A. Solanaceae. Plants in Solanaceae, like tomatoes and potatoes, Leguminosae, and Rosaceae often have pollen that remains viable for several months. Poaceae, B, which includes cereals like rice and wheat, typically have short pollen viability, often less than 30 minutes. Liliaceae, C, and Orchidaceae, D, also show relatively short viability periods. Hence, Solanaceae stands out for its extended viability duration. What feature of pollen grains allows their preservation as fossils? A. Presence of cytoplasm. B. Hard in time. C. Sporopollenin-rich exine. D. Lipid coating. Correct answer is C. Sporopollenin-rich exine. Explanation. The correct answer is C. Sporopollenin-rich exine. The exine is the outer layer of pollen, composed of sporopollenin, which is highly resistant to microbial decay, oxidation, and enzymatic degradation. This makes pollen grains one of the most durable structures in the plant kingdom, allowing them to be preserved in fossil form. Cytoplasm, A, and lipid coating, D, degrade quickly and are not fossil preservable. In time, B, is the inner layer made of cellulose and pectin, which also degrades over time. Only the exine ensures long-term fossil preservation. Where does microsporogenesis occur? A. Microsporangium. B. Ovule. C. Petal. D. Style. Correct answer is A. Microsporangium. Explanation. The correct answer is A. Microsporangium. Microsporogenesis is the process of formation of microspores from pollen mother cells via meiosis, and it takes place inside the microsporangia of the anther. Ovule, B, is part of the female reproductive structure. Petal, C, and style, D, are not involved in microspore formation. Thus, microsporangium is the correct and exclusive site of this process. Why are pollen grains from Parthenium have a health concern? A. They are allergens. B. They lack nutrients. C. They infect flowers. D. They are genetically modified. Correct answer is A. They are allergens. Explanation. The correct answer is A. They are allergens. Parthenium hysterophorus releases airborne pollen that is highly allergenic, leading to respiratory issues like bronchitis and skin reactions in sensitive individuals. Option B, lack nutrients, is irrelevant because pollen isn't consumed for nutrition. Option C, infect flowers, is incorrect as Parthenium pollen doesn't infect but pollinates. Option D, genetically modified, is incorrect. Parthenium is not a GMO issue. Allergic response is the specific health concern here. Why is sporopollenin considered an evolutionary advantage? A. It helps in fertilization. B. Provides durability and fossil record. C. Aids in flower development. D. Enhances color. Correct answer is B. Provides durability and fossil record. Explanation. The correct answer is B. Provides durability and fossil record. Sporopollenin makes pollen exine incredibly tough, allowing it to resist decay and endure for centuries, even as fossils. This durability gave reproductive success across variable environments. Option A, helps in fertilization, is incorrect. It's the pollen tube that aids fertilization, not sporopollenin. Option C, aids in flower development, and D, enhances color, have no link to this polymer. The evolutionary benefit is in structural resilience and historical traceability. Why might plants with short pollen viability need precise pollination timing? A. To form fruit early. B. To ensure ovary development. C. To avoid pollen death before reaching stigma. D. To attract pollinators. Correct answer is C. To avoid pollen death before reaching stigma. Explanation. The correct answer is C. To avoid pollen death before reaching stigma. In plants like rice and wheat, pollen grains are viable for just 30 minutes to a few hours. Thus, pollination must occur within this window to ensure fertilization. Option A, form fruit early, and B, ovary development, depend on successful fertilization, not timing per se. Option D, attract pollinators, is unrelated to pollen viability. Timely pollination ensures pollen can complete its function before losing viability. How does tapetal binucleation benefit microspore development? A. Increases temperature. B. Enhances nutrient synthesis. C. Protects exine. D. Forms endothecium. Correct answer is B. Enhances nutrient synthesis. Explanation. The correct answer is B. Enhances nutrient synthesis. Binucleate tapetal cells are metabolically more active and synthesize a wide range of enzymes and nutrients essential for pollen development. Option A, increases temperature, is incorrect. Temperature regulation is not a function of tapetum. Option C, protects exine, is not a direct function, although tapetum does supply materials for exine. Option D, forms endothecium, is false. Endothecium is a separate anther layer. Binucleation is key for the nutritional role of tapetum. Why is pollen tube growth always through germ pores? A. It's random. B. Germ pores are weak areas. C. Exine is absent there. D. More cytoplasm is there. Correct answer is C. Exine is absent there. Explanation. The correct answer is C. Exine is absent there. Germ pores are specialized regions where the tough sporopollenin-rich exine is absent or reduced, allowing the pollen tube to emerge easily and penetrate toward the ovule. Option A, random, is incorrect. Pollen tube emergence is a highly directed process. Option B, weak areas, is vague and not technically accurate. It's not about weakness but structural adaptation. Option D, more cytoplasm, is irrelevant to emergence. The absence of exine makes germ pores the ideal emergence point. Why is the intine softer than exine? A, to reduce weight. B, made of cellulose and pectin. C, allows water loss. D, for germination energy. Correct answer is B, made of cellulose and pectin. Explanation. The intine is the inner layer of the pollen wall, and it is softer than the exine because it is composed of cellulose and pectin, materials that are flexible and allow for expansion during pollen tube growth. This softness is essential for the emergence of the pollen tube during germination. Option A is incorrect because the softness isn't related to reducing weight. Option C is misleading as intine doesn't function to allow water loss. Option D, while vaguely associated with germination, doesn't accurately reflect the structural reason for its softness. How does dehydration affect pollen tetrads? A, strengthens them. B, kills them. C, separates individual microspores. D, triggers mitosis. Correct answer is C, separates individual microspores. Explanation. During microsporogenesis, dehydration causes the pollen tetrads, a group of 4 microspores, to separate into individual microspores. This is a preparatory step for each microspore to develop into an independent pollen grain. Option A is incorrect because dehydration does not strengthen the tetrads. It weakens the callose wall that holds them together. Option B is wrong as dehydration is a normal developmental process, not lethal. Option D is irrelevant here. Mitosis has already occurred before this separation. Why might exine architecture vary between species? A, different food reserves. B, species-specific identification and dispersal adaptation. C, vascular needs. D, all pollen grains are identical. Correct answer is B, species-specific identification and dispersal adaptation. Explanation. The exine has species-specific sculpturing patterns that play a role in recognition during pollination and also reflect adaptations to different dispersal methods like wind or insect pollination. This variation ensures successful reproduction. Option A is unrelated. Exine has no food storage role. Option C relates to plant transport systems, not pollen wall structure. Option D is factually incorrect. Pollen grains show significant diversity across species. Which storage technique allows long-term pollen preservation for breeding? A, oven drying. B, ethanol fixation. C, storage in liquid nitrogen. D, air drying. Correct answer is C, storage in liquid nitrogen. Explanation. Pollen is best preserved for long-term use in breeding programs through cryopreservation in liquid nitrogen at -196 degrees Celsius. This ultra-low temperature halts all metabolic activity, maintaining viability for years. Oven drying and air drying cause damage to pollen viability due to high temperature or desiccation. Ethanol fixation is used for preserving cellular structures, not maintaining viability for fertilization. If a plant's pollen is non-viable after 20 minutes, what is essential for fertilization? A, long styles. B, no pollinators. C, synchronization of stigma receptivity and pollen release. D, high light intensity. Correct answer is C, synchronization of stigma receptivity and pollen release. Explanation. When pollen has a very short viability period, it is crucial that the stigma becomes receptive precisely when the pollen is released. This synchronization ensures successful fertilization. Long styles, A, could delay fertilization, making them counterproductive in this case. No pollinators, B, would reduce fertilization chances further. High light intensity, D, is not a determining factor in such synchronization. Which pollen feature would benefit a species pollinated by wind? A, heavy and sticky grains. B, light and smooth grains. C, brightly colored grains. D, strongly scented grains. Correct answer is B, light and smooth grains. Explanation. Wind-pollinated species produce light and smooth pollen grains, which are easily carried through the air. Heavy or sticky grains, A, are adapted for animal pollination. Bright colors, C, and scents, D, are also typical of biotic pollination strategies involving insects or animals and are irrelevant for wind dispersal. How might pollen supplements help athletes, as claimed? A, increase oxygen in muscles. B, cure diseases. C, provide dense nutrients and antioxidants. D, stimulate adrenaline. Correct answer is C, provide dense nutrients and antioxidants. Explanation. Pollen supplements are rich in proteins, vitamins, and antioxidants, which are claimed to boost energy levels and aid in recovery, though scientific support is mixed. They do not directly increase oxygen in muscles, A, nor do they cure diseases, B, or stimulate adrenaline, D. These claims are exaggerated and not supported by clinical evidence. Why is knowledge of microsporogenesis crucial in plant breeding? A, helps identify fruit color. B, ensures insect pollination. C, allows artificial hybridization via controlled pollen. D, predicts flower size. Correct answer is C, allows artificial hybridization via controlled pollen. Explanation. Microsporogenesis is the process of pollen formation, and understanding it enables breeders to control and collect viable male gametes for artificial hybridization. This is vital for developing new plant varieties. Fruit color, A, and flower size, D, are related to other traits. Insect pollination, B, is natural and not directly influenced by knowledge of microsporogenesis. What could be a result of a defective tapetum in a plant? A, more petals. B, larger leaves. C, sterile pollen. D, early seed formation. Correct option is C, sterile pollen. Explanation. The tapetum provides nutrients and enzymes required for pollen development. If it is defective, pollen grains may fail to mature properly, leading to sterility. More petals, A, and larger leaves, B, are unrelated to tapetum function. Early seed formation, D, requires successful fertilization, which is compromised if pollen is sterile. Why would a palynologist study exine patterns in ancient rocks? A, to find fossils. B, to study historical plant biodiversity. C, to analyze soil erosion. D, to observe animal migration. Correct answer is B, to study historical plant biodiversity. Explanation. Palynologists examine fossilized pollen with distinct exine patterns to reconstruct ancient plant communities and climate. While these are technically microfossils, the primary goal is not fossil hunting, A, but ecological and evolutionary insights. Soil erosion, C, and animal migration, D, are not directly related to pollen morphology in rocks. Which modern application uses stored pollen for future use? A. Vaccine development. B. Animal cloning. C. Crop improvement programs. D. Food coloring. Correct answer is C. Crop improvement programs. Explanation. Stored pollen is used in breeding programs to create hybrids with desired traits like disease resistance or higher yield. It is not used in vaccine development, A, or animal cloning, B, which involve different biological materials. Pollen is also not a source of food coloring, D, although it may contain pigments. Assertion. The tapetum is the innermost layer of the microsporangium and is critical for nourishing developing pollen grains. Tapetal cells are metabolically active and often possess more than one nucleus. Reason. Tapetal cells undergo amitotic division during microsporogenesis, leading to multinucleation. A. Assertion is true, reason is false. B. Assertion is false, reason is true. C. Both assertion and reason are true but reason is not the correct explanation of assertion. D. Both assertion and reason are true and reason is the correct explanation of assertion. Correct option is C. Both assertion and reason are true but reason is not the correct explanation of assertion. Explanation. The assertion is correct because the tapetum is indeed the innermost layer of the microsporangium and plays a vital role in nourishing the developing pollen grains. It is metabolically active and commonly becomes binucleate. However, the reason is incorrect in its mechanism. Tapetal cells do not undergo amitotic division. Rather, their binucleate nature results from either nuclear division without cytokinesis or endomitosis. Hence, while both statements are true, the reason does not correctly explain the assertion, making option C the best choice. Option D is incorrect because the explanation is faulty. Option A and B are eliminated since neither statement is false. Assertion. A typical anther in angiosperms is bilobed and dithecous. Reason. Each lobe contains two microsporangia, which later develop into pollen sacs. A. Assertion is true, reason is false. B. Assertion is false, reason is true. C. Both assertion and reason are true but reason is not the correct explanation of assertion. D. Both assertion and reason are true and reason is the correct explanation of assertion. Correct answer is D. Both assertion and reason are true and reason is the correct explanation of assertion. Explanation. The assertion accurately reflects that a typical anther in angiosperms is bilobed and dithecous, meaning each lobe has two laca or microsporangia. The reason clearly supports this by stating that two microsporangia develop in each lobe, which eventually form pollen sacs. This structure while explaining anther anatomy. Because both the assertion and reason are correct, and the reason logically explains the assertion, option D is correct. Option A and B are invalid because neither of the statements is false. Option C is incorrect since the reason does serve as an explanation. Assertion. Microspores produced in tetrads separate from each other before developing into pollen grains. Reason. Dehydration of the anther causes the separation of microspores from the tetrad. A. Assertion is true, reason is false. B. Assertion is false, reason is true. C. Both assertion and reason are true but reason is not the correct explanation of assertion. D. Both assertion and reason are true and reason is the correct explanation of assertion. Correct answer is D. Explanation. The microspores are formed in tetrads during microsporogenesis, but these separate from one another due to dehydration of the anther as it matures. This makes the assertion and reason both factually accurate and the reason directly explains why the microspores dissociate. Thus, option D is correct. Option C is wrong because the reason does explain the assertion. A and B are eliminated because neither statement is false. Assertion: sporopollenin in the exine of pollen grains makes them extremely durable. Reason: sporopollenin is highly resistant to biological and chemical degradation, including action by enzymes, acids, and bases. A. Assertion is true, reason is false. B. Assertion is false, reason is true. C. Both assertion and reason are true but reason is not the correct explanation of assertion. D. Both assertion and reason are true and reason is the correct explanation of assertion. Correct option is D. Explanation: the assertion is true. Sporopollenin in the exine makes pollen grains extremely durable. Sporopollenin is the most resistant organic substance known and can withstand extreme physical and chemical conditions. The reason accurately supports this, explaining the resistance to acids, bases, and enzymes. Since the reason directly clarifies the durability mentioned in the assertion, option D is correct. Option C would be wrong because the explanation is indeed valid. A and B are incorrect because both statements are true. Assertion: pollen grains are usually shed in the 2-celled stage in most angiosperms. However, in some species, they are shed in the 3-celled stage. Reason: in the 3-celled stage, the generative cell divides mitotically into 2 male gametes before the pollen grain is shed. A. Assertion is true, reason is false. B. Assertion is false, reason is true. C. Both assertion and reason are true but reason is not the correct explanation of assertion. D. Both assertion and reason are true and reason is the correct explanation of assertion. Correct option is D. Explanation: the assertion states that over 60% of angiosperms shed pollen in the 2-celled stage, and the rest in the 3-celled stage. The reason is also valid. In those that reach the 3-celled stage, the generative cell divides mitotically to form 2 male gametes before the pollen is shed. Since the reason correctly explains the assertion, option D is the right choice. Option C fails because it denies the causal relationship that clearly exists here. A and B are ruled out since both statements are factually accurate. Assertion: a typical microsporangium has 4 distinct wall layers. Reason: the outer 3 layers of the microsporangium help in pollen grain nourishment. A. Assertion is true, reason is false. B. Assertion is false, reason is true. C. Both assertion and reason are true but reason is not the correct explanation of assertion. D. Both assertion and reason are true and reason is the correct explanation of assertion. Correct option is the assertion is true, reason is false. Explanation: the assertion is correct because the microsporangium has 4 wall layers: the epidermis, endothecium, middle layers, and tapetum. However, the reason is false. It wrongly states that the outer 3 layers nourish the pollen grains, when in fact only the tapetum performs this function. The outer layers are responsible for protection and help in anther dehiscence. Hence, option A is correct. Option D is incorrect because the explanation is faulty. C is wrong because the reason is not even factually correct. Option B is also eliminated. Assertion: pollen grains can be preserved for years when stored in liquid nitrogen. These preserved grains can be used for crop breeding. Reason: Pollen grains remain viable indefinitely if kept at room temperature and low humidity. A. Assertion is true, reason is false. B. Assertion is false, reason is true. C. Both assertion and reason are true but reason is not the correct explanation of assertion. D. Both assertion and reason are true and reason is the correct explanation of assertion. Correct option is A. Assertion is true, reason is false. Explanation. The pollen grains can be preserved for years using cryogenic storage in liquid nitrogen at 196 degrees C and used in crop breeding. Thus, the assertion is true. The reason is incorrect because pollen grains do not remain viable indefinitely at room temperature. In fact, in cereals like rice and wheat, viability is lost within 30 minutes. Therefore, the correct answer is A. Option D and C are wrong because the explanation is invalid. Option B is ruled out because the assertion is not false. Assertion. The process of microsporogenesis ensures genetic variation in pollen grains. Reason. Microspore tetrads are formed through mitotic division of sporogenous tissue. A. Assertion is true, reason is false. B. Assertion is false, reason is true. C. Both assertion and reason are true but reason is not the correct explanation of assertion. D. Both assertion and reason are true and reason is the correct explanation of assertion. Correct option. A. Assertion is true, reason is false. Explanation. The assertion correctly states that microsporogenesis results in genetic variation, as the process involves meiosis, leading to recombination and formation of haploid microspores. However, the reason is false. It incorrectly claims that the microspore tetrads are formed by mitotic division. The meiosis occurs in pollen mother cells to produce microspores. Hence, the correct answer is A. Option D and C are incorrect due to the flawed reasoning. Option B is ruled out because the assertion is valid. Assertion. Pollen grains show great diversity in their architecture such as shape, size, color and ornamentation. These features are species-specific and help in identification. Reason. The exine of pollen grain is made up of cellulose and pectin, allowing intricate patterns. A. Assertion is true, reason is false. B. Assertion is false, reason is true. C. Both assertion and reason are true but reason is not the correct explanation of assertion. D. Both assertion and reason are true and reason is the correct explanation of assertion. Correct option is the assertion is true, reason is false. Explanation. The assertion is factually correct. Pollen grains exhibit a wide variety of features, such as shape, size, and surface patterns, which are species-specific and aid in identification. However, the reason is false. The exine, responsible for these patterns, is made of sporopollenin, not cellulose and pectin. Cellulose and pectin make up the intine, the inner wall. Hence, option A is correct. D is wrong because the reason is incorrect. B and C are also not valid because the assertion is not false. Assertion. Inflorescence and floral primordium development begins much before the flower is visible. This includes structural and hormonal changes. Reason. These early changes help the flower undergo pollination after fertilization. A. Assertion is true, reason is false. B. Assertion is false, reason is true. C. Both assertion and reason are true but reason is not the correct explanation of assertion. D. Both assertion and reason are true and reason is the correct explanation of assertion. Correct option is C. Explanation. The assertion is valid. Floral development begins long before the flower visibly appears, triggered by hormonal and structural changes that lead to formation of inflorescences and floral buds. The reason is incorrect because it suggests that these changes help in pollination after fertilization, which is factually wrong. Pollination precedes fertilization. Hence, while both statements are true independently, the reason is not the correct explanation for the assertion, making option C correct. Option D is incorrect as the cause-effect relationship is invalid. A and B are eliminated since neither statement is false.